So we're going to go through actually every single block in this page because it's such a huge page. It's very dense with material. I know I said that a few slides ago, but I mean conceptually it's not that hard, right? But when we start going into the nitty gritty, it might seem a little more challenging than it does at first. Okay. So you have two regions on any sort of antibody. You have the constant region and FC, and then you have the AB region, which is the antibody region. At least that's how I remember it. Okay. So the antigen ends up binding here, in the antigen binding region, AB. Complement binds right here. Now that's huge because I've seen questions where they literally have things like A, B, C, down here D or something. And they ask you, what does complement bind? And if you're not clicking here, you're not getting that right. Antigens always bind here, by the way. It's the antigen binding site, AB. Okay, so before starting all of that, there's three different terms here that they don't go into too much, but you will see on exams. Isotype, allotype, and idiotype. Isotype is IgG versus IgM, which is of course determined by the heavy chain. Okay, allotypes is just differences in different people's, IgGs versus, IgMs and IgAs. What's mine look like? What does yours look like? But we're all the same antibody here. And of course, idiotypes have different, the idiotype is characterized by this region right here, where the antigen actually ends up binding. All right. So, yeah, you have this heavy chain right here, and then you have the light chain on the side. What else? The FC region of IgM fixes complement. We already talked about that. Heavy chain contributes to the FC and FAB regions, which is true. Constant region. This is the antigen binding region. You're going to get questions possibly on, the light chain contributes to. You'll have a choice between, the variable region. You have a choice with the constant region, and you have to say light chain is only with the variable region. Okay. FAB, antigen binding, right. Determines idiotype. Haha, exactly. Very specific, 1 for 1. Constant region determines the isotype. IgG versus IgM. That makes sense. It'll make more sense maybe in the next page where you can see how different all these things are. IgM with the J chain and pentamer structure. There it'll make more sense instead of just looking at it like this. Okay, here's the real thing. Antibody diversity. Okay, and then antibody specificity. So the question that everyone has is, okay, great, how do you make a zillion different types of antigen binding regions in order to grasp different types of antigens? And then the next question is, well, that's great that this T cell is secreting cytokines to get it to class switch, but how do you actually class switch? Let's open up some really cool things. So here we have a VJ region. Now, if you remember, VJ is on the light chain and VDJ is on the heavy chain. So here's how you're able to kind of change what happens on the antigen binding site in the antigen binding region. You have all these sequences. This is all DNA, by the way, and you have all these little regions coding for, variable region type A or variable region type B, type C. This is the J region. And you can just imagine that if this were the heavy chain, you'd have a D right here in the middle between the variable and the J section. Well, so what ends up happening? Well, you've got your RAG, you have your RSS sequences, okay? Everything gets snipped and then gets recombined. And you can see how your variable subtype B and your variable subtype C is eliminated. And here your variable A region is connected to the J sequence. And so this is an example of VDJ recombination. Of course, if we were working with a heavy chain, you would see how there would be a D sequence. It would be VDJ recombination. This is also very high yield. TDT adding nucleotides. So one more time, what's going on here? You have these sequences tagged by RAG, which brings your two regions together, cuts out the unessential region, and then with TDT adding nucleotides, they're joined together. And you have a specific antigen binding region. There's way more than just A, B, or C different types of variable change. There's a zillion different types. I just put 3 there because I obviously can't draw a zillion different things. Okay. But there's more. So next we have hypermutation. So what this has to do is you go within the variable region, right, which is right here, And when you actually look, you have hypervariable locations called the complement determining regions. Now, you have activated cytosine deaminase, which ends up switching the hell out of these things and giving them a whole bunch of different, flavors, you can say, to really perfectly match something. And it's through this somatic hypermutation that you end up getting 1-for-1 matches between antibody and antigen. So, again, this is within the variable region. You have these CDR regions, and these things are responsible for somatic hypermutation. In fact, if we kind of look here, you can assume that these little bands here is what we're talking about. Okay. The last thing we need to talk about right now is isotype switching. Isotype switching, I'm going to go back to the same page. Give me 1 second. Isotype switching. Remember, isotype, IgM, IgG, IgD, Ig whatever. Isotype switching is pretty simple. You have a strand here of DNA. Here's your coding sequence, right? And we've already went through VJ and VDJ, recombination and how all those things get switched around to allow for more variability in terms of antigen recognition. But what happens if you want to do something more IgA? What if you want to do some gut dependent stuff? Or what if you've already done your primary response, IgM, and now you're looking to do IgG? What do you do? What's going on? Well, it's actually pretty straightforward. It's pretty easy. So there's a great video, and I guess I'll post the link to it because just writing this down might be a bit tough. And all credit for this diagram goes to one of these videos. I forget which one it is. Probably this one. Anyway, so the idea here is that here is your sequence of your coding sequence, right? And in order to switch from IgM, which is always made first, and you'll notice here that this is IgM, in order to switch from IgM to literally anything else, IgG, IgE, IgA, what happens is a process of 2 cuts. So you of course cut here after your VDJ segment and before your constant segment. So you cut here, and then let's say you want IgE. You want to class switch to IgE. Well, you cut here, and then you cut here. And then this section here is gone, and what's annealed is this IgE thing. And this attaches to here. Now you just have IgE. You might be wondering, what happened to the IgA? Well, the IgA stays there. The IgA, this segment doesn't move. But what gets transcribed is your IgE, because the first thing here, and then it stops. Well, you would say, okay, well, what if I just want IgG? I want to go from IgM to IgG. Okay, well, you cut here, and you cut here. And now this IgG goes forward. I'm not doing justice to the video, so hopefully you'll watch that. Okay, so what can antibodies do? Well, they can neutralize, so it can't bind to anything. You can promote phagocytosis, or as we'll show later on, you can activate complement, which just beats the hell out of everything. And remember, if there's no MAC complex, you'll end up getting stuff like Neisseria, but that's for another time.